Okay, good morning. Welcome to IPMWORKS, e-learning modules. My name is Ramon Albages from Agrotechnic Center at the University of Lleida. This video is part of the Module 1, Agroecosystem Concepts and Theory, Holistic Approach to IPM. And I will present the chapter number 7, a case study about protected crops. Let's see some common characteristics of protected crops that influence the application of IPM. The products from greenhouse productions have a very high market value. Vegetables and ornamentals can be for local consumption or for exportation. A second characteristic is there is a high demand for high quality standards. The third characteristic, there is a very active international trade of plant material. So there is a high risk of introduction of invasive exotic pests. And fourth characteristics, protected crops have a very warm environment, and this leads to a fast development of pest populations. It could therefore be expected that pest control in greenhouse crops would be exercised mainly by pesticides, especially in ornamental crops. However, greenhouse production is the agricultural sector with the greatest application of biological control. Let's see which factor in the last 50 years have made that possible. Let's talk about the importance of the landscape surrounding greenhouses. Let's compare in the following pictures where greenhouses are inserted. The proportion of agricultural land under plastic or under glass, the proportion of agricultural land uncovered, so in the open air, the proportion of non-crop habitats, the proportion of urban infrastructure, houses, roads, railways, waterways, etc. Here you can see a picture in Liguria, in the north of Italy, 800 millimeters of precipitation. And you can see that some plastic greenhouses, some also glass houses, some urban areas, some non-crop habitats, some open-air agricultural land. Similar landscape you can see in Catalonia, about 600 millimeters on the coast of Catalonia. There are many plots in open air, but also some plastic and glass house, some non-crop habitats. And we go to the south of Spain, in Murcia. It's lower precipitation, but especially with very dry summer. We have also here open air land, some greenhouses, and also in some places, some non-crop habitats. Let's see in the south of Italy, in Sicily, in the area of Catania, 500 millimeters of precipitation, but very dry springs. Most of the land is covered by plastic, and there is very, very little surface of open air vegetables. Also in Crete, we have here a very big concentration of plastic houses, and let's see in Almeria, most of the land is covered by plastic. This is called sometimes the plastic sea, very low precipitation. So let's see some evolution of pest control in European Union greenhouses. Pest control in greenhouses started with pesticides, 20th century. You can see here, ornamental glass house, here some vegetables, also some tomatoes, and treated with pesticides. At the beginning, pesticides were used for pest control, but some future led to move to other non-pesticide methods to control pests. First, an increasing sensitivity of consumers to toxic pesticide residues on food. Second, health risk for greenhouse workers. Third, awareness of the damage caused by chemicals on the environment. Fourth, an increasing insecticide and pesticide resistance in several pests. And the last, more and more pesticides forbidden. So this led to substitute pesticide by biological control. Biological control was initiated in Northwestern Europe, in England and the Netherlands in the first half of the 20th century. You have here some natural enemies used at that time. This is Encarsia formosa, a parasitoid of greenhouse whitefly. You can see here some nymphs of greenhouse whitefly. The black nymphs are parasitized by Encarsia formosa. And here we can have a predatory mite attacking a herbivorous mite, Titanicus urtice. Which are the limitants of IPM development in protected crops? Climate conditions inside greenhouses are not fully suitable for the natural enemies that were used at the beginning. High pest and virus pressure on greenhouse from outside. Arthropods coming inside greenhouse. Lack of commercial providers of natural enemies in the region. In the Mediterranean, first, there were no companies selling natural enemies. The companies were in the Netherlands or in England. Multiple introduction of exotic pests and vector diseases. Some examples. Bemisia tabaci, it's an excellent vector of the tomato yellow leaf curl virus. Then we had Frankiniella occidentalis, a thrips. It's also a vector, another virus. And also we had Thrips palmi, but the Thrips palmi was not established, and to my knowledge is not still established in the European Union. Liromycia hodobranchis and Liromycia trifoli. Also we had Liromycia sativa. You can see here the Liromycia hodobranchis, Liromycia trifoli. Here also the adults. Titanicus evansi and Titanicus turkestani. Here. Tuta absoluta. Here you can see the adult. You can see the damage on tomatoes. And we expect to have in the coming years, perhaps, Bactericeri coccarelli. That's an important pest. Now, let's see which is the IPM in the landscape. Here, there is a representation of the cycle of arthropods coming into the greenhouses or leaving greenhouses and going to the outside vegetables or wild vegetation. And along the year, you can see that all the arthropods can go into greenhouses and leave the greenhouses. And this means that they can colonize the outside, the inside, through the vents of the greenhouses. At the beginning, we thought that the insects left the greenhouses, especially in summer, and come into greenhouses in winter. But when we started to monitor the flights of the insects from inside to outside the greenhouse, or from outside to inside of the greenhouse by means of glued traps, we could see that this hypothesis was not true. The flying insects are able to live and to go into the insects along all the year. You can see here that in blue, you can see the number of insects coming into greenhouses, and in yellow, the insects leaving the greenhouses. You can see that every week there are insects leaving and some other insects coming into greenhouses. So there is a continuous movement of insects between inside and outside greenhouses. But the manipulation of the landscape around greenhouses is not always feasible. Land in artificial areas is often expensive, so we cannot devote some part of the landscape outside to install refuges for natural enemies. Fallows are not practiced outside because the land is very expensive. Sometimes there are no areas with natural habitats. You can see here in Almeria. Greenhouse areas are placed in dry regions with a very poor natural vegetation. And greenhouse vents are usually or are often screened. You can see here, we are inside the greenhouse, and perhaps you cannot see, but this is a net. This is a net. This is a net that prevents that the insects come into greenhouses or leave the greenhouses. And so natural enemy in these conditions are not able to go into greenhouses. We started research trying to see which is the influence of the this is the influence of the percentage of the coverage around the greenhouse with the detection of the first detection of the predatory Macrolophus pygmaeus, a predator. We can see as we have more vegetation outside, from 0% to 24%, the precocity is higher, because this is the first detection of the predator in the greenhouse. More cover outside, and earlier is the detection of the first predators. Here, some other conclusion of other French team working in the south of France. And the first stage suggests that maintenance of large areas of semi-natural habitats within a radius of 300 meters is important to ensure colonization of protected crops by predatory mealybugs, in particular with two predators, Macrolophus and Sisyphus. On the contrary, the proximity of orchards to vegetable plots or greenhouse reduces crop colonization by midges, and this could reflect either sink effects due to intensive management practice in orchards, particularly the pesticide treatment. Okay, thank you for your attention.